Okay, now we move to Vincent Mariaule. So Vincent, he's member of the MIA team working on the microbiome-host interaction, and today he will present recent results which was published into 2024-25 about diet intervention in dog. Please, the floor is yours.
So, thank you for the nice introductions, and thank you very much for giving me the opportunity to present such results in the IBD days. So, as it was nicely introduced by thank you. In the presentation by Emmanuel, I think now it's clear for everyone how important is the gut microbiome through health. And indeed, you have a lot of studies that demonstrate this role in health and disease. When we talk about the gut microbiota, it's true that it's a quite complex thing, and although we are getting more information on mechanical aspect and so on, there is a lot of things we are still not aware of, and we still to investigate. And for example, when we look at the microbiota, we have different parameters that can define it and that can affect how it interacts with the host. And the basic thing we can think about is the composition, its diversity, but also its functionality, production of enzyme and so on, synthesis of, for example, vitamins and so on. The resilience, how stable can be the gut microbiota to perturbations. But also an aspect of which is important is the metabolites that are produced by the gut microbiome. I said previously, one of the main factors that can change, in fact, the gut microbiome is diet. As not only is affecting the composition, but also the functions of the gut microbiota. And this modification can be for the good, towards health, and we saw examples, for example, about Mediterranean diet or other diets, but also to the worse. And for example, we have the so-known Western diets that have been also associated in humans with IBD disease. So usually, studies have also demonstrated that this dysbiosis of the gut microbiota is usually associated to disease. So we talk about humans, but that's also true for animals. And here I will talk about more about inflammatory enteropathies in dogs. So which mainly characterized by digestive clinical signs that persist more than 3 weeks, and can be also accompanied by extradigestive disorders that represent about 1 or 2% of the total clinical reviews, and no effective treatment so far. So when it comes to inflammatory enteropathies, the actual classification relies on therapeutic response. And this leads to 3 different categories. A first one that is called food responsive enteropathy, where actually we are modifying the diet of the dog, and we can see some improvements. We then have a strategy based on microbiota modulations, responsive enteropathy. And then the third class was close to the, let's call it inflammatory bowel disease, subdivide into immunomodulators, responsive enteropathies, and also refractory enteropathies. So, so far, the etiology of such pathologies remain unknown, yet they seems to result from the interaction from different factors, including genetic, environmental, immune, but also microbiota interactions. And if we look a bit more on the patients, we see functional dysregulations that are characterized by gut microbiota dysbiosis, with the increase of actinomycetota and pseudomonadota, and a decrease of bacillota and bacteroidota. We also saw metabolic dysregulations, with increase of primary bile acids and decrease of secondary bile acids, and increase of dysregulation of the tryptophan metabolisms, and decrease of short-chain fatty acids. But also, we have a disruptions of the proteolytic activity and homeostasis that is represented by an increase of serine proteases activity and a decrease of the natural inhibitor, the serine protease inhibitor. So many studies that study the impact of diet on CA in dog are cross-sectional, meaning that see to describe how the diet, a unique diet on dogs. Mainly, we have two trends for diets in dogs, which called natural diets versus industrial diets. So the first one, the natural diet, is composed of fresh, unprocessed, or minimally processed products. However, they seem to be unbalanced nutritionally for the dogs. Also, most of the studies reported at the microbiota level an increase, a high abundance of Fusobacteriaceae and Bacteroideaceae family. In the opposite, the industrial diet, it's more manufactured diet, which encompasses a minimum quantity of starch for the extrusion process and is designed to be nutritionally balanced. At the microbiota level, so far, we can see a high abundance of Bacillota. So let me rephrase. So why to be interested in dog, basically? Well, several reasons for that. First, they have spontaneous inflammation pathologies in dogs. They share the same environmental exposures as humans. That could be very interesting as a model. They have a binary habits for eating, either natural or industrial. And also, that is really interesting, especially for diet interventions, is that we can carry out clinical trials in complex environments. So the objective of our research was to determine, to analyze the impact of dietary change on the microbiota and health response in semi-stray dogs from Tunisia. To do so, we first construct a cohort of semi-errant dogs that were well characterized, so they were clinically healthy, they were devoid of any intestinal parasite, they were not vaccinated or dewormed, they had not received any antibiotic treatment for at least 6 months, obviously to not have any impact on the gut microbiota. And why using semi-errant or semi-stray dog is first because of their eating, which are uncontrolled eating that is based on raw meat, leftover meals, corpse, prey. But also they have the advantage that we can maintain them in their environment. They have a higher control of the dietary impact. So this is the design that we have adopted. So we have our cohort of semi-stray dogs that were divided into groups. First, the group 1 that represents our control group, that they kept their self-nourished food habits. And then the second group was dedicated to the dietary intervention, with first a gradual transition over 10 days and towards the industrial diet that we keep for a month to be sure we can observe any effects for this new diet. Sorry. Also to precise that at the beginning of the dietary interventions and at the end, we were collecting 2 samples. From these 2 samples, we used them for several analyses. The first one was to determine the gut microbiota composition. So here we have an overview of the methodology we use to obtain microbiota profiling based on 16S sequencing. But we also use tools to perform a functional analysis, which allowed us to determine the bile acid profiles, such as acid fatty acid profiles, but also the proteolytic activities. We also collect some fresh bloods to monitor glucose concentration and also C-reactive protein for inflammatory markers. So the first thing we have looked is the fecal proteolytic activities. So unfortunately, you can see here, but on the graph on the left, you see the total proteolytic activity, fecal. So on the left, you have the self-nourished group, and on the right, you have the industrially nourished group. What you can see is that on the self-nourished group, there is no significant difference, which is great. And when it comes to the interventions, we clearly see that after 40 days, we have a 50% decrease of the total proteolytic activities. Then, on the right, we try to characterize a bit more about these proteolytic activities in terms of family, protease family. So for that, we use specific inhibitors, and the interesting thing is that we are using PMSF that appear in blue, which is a broad spectrum inhibitor for serine protease family. We can see that we have a significant decrease of the proteolytic activity, meaning that this family, so the serine protease family, represent the most increased protease family in the total activities. Then we wanted to go a bit further on the characterization of the serine protease, and we use a specific substrate to go a bit further on the profiling. And as you can see, we saw differences in 4 different types of activities. So again, for the control group, we don't see any change that is significant, but on the industrial part, we see some change. So we should see a strong decrease in trypsin, neutrophilic elastase, and PR3 activities at the end of the diet change. And we also see an increase of the chymotrypsin activities on the top right, as you can see. That will be some interesting parts later. No. Great. No. Good idea, but no. Okay. Then we move on the analysis of short-chain fatty acids, and again, control of nothing, but once change for the industrial diet, we see that we have a significant increase on 3 major short-chain fatty acids, which are acetate, isobutyrate, and isovalerate. We then analyze the bile acid profile. Same thing, nothing for the control group, but when it comes to the industrial nourished group, we can see that we have the primary bile acids, so the cholic acid on the left and the deoxycholic acid, chenodeoxycholic acid here, that are significantly decreased, so by a factor of 22.5 for the cholic acid and a factor of 14 for the chenodeoxycholic acid. But more importantly, we see that we have a significant increase for 2 of secondary bile acids, which are beneficial bile acids. On the left, so the iodo-deoxycholic acids and the litocholic acids. Now we have a look on the composition of the intestinal microbiota. So between our 2 groups, we kept the same main film analysis, but when we look at the industrially nourished one, we clearly see a change in the composition of intestinal microbiota that is characterized by an increase of Bacteroidota and Fusobacteriota, and a decrease of Pseudomonadota and Actinomycetota. When we go a bit deeper in the analysis, we can see that whether we observe the richness of the gut microbiota or the diversity by the Shannon or Simpson index, we can see that we have an increase of bacterial abundance and richness in dogs after being put on industrial diets. We go a bit deeper and look for the beta diversity. Still, on the control group, no difference can be seen. However, after the food transition, we are clearly difference between before and after the interventions. We look now at the genus level for the gut microbiota, and we also highlighted that we have an increase of beneficial bacteria for the health, such as Bacteroidotes, Butyriccoccus, Lactobacillus, and Faecalibacterium. We then try to see if we could find any correlation between the increased bacterial genus and pharmaceutical functions related to SCFA, bile acids, and proteolytic activity, and what we could find is that we have a positive correlation between bacteroidetes abundance and chymotrypsin activities. Remember that I told that we saw this increase of chymotrypsin activity. So now we have kind of correlations, and also we saw that we have a negative correlation between Peloclostridium and the production of isovalorates. Keeping in mind that this can have obviously an impact on health and well-being of the dogs. So in conclusions, we perform a transition to industrial foods on dogs, and we show that we have an increase of microbial diversity and richness in dogs having subjected to the diet change. We have an increase in the production of SCFA, and we have a conversions from primary bile acids to secondary bile acids. We also saw a decrease in proteolytic activities, notably serine protease. So this experiment study is a proof of concept that the diet has the capacities to modulate functionalities and composition of intestinal microbiota, and that the diet could be an alternative approach as a lever for the prevention and treatment of digestive diseases. Now, as a perspective, we thought to analyze the potential of diet, this time to restore functional disorders associated to chronic enteropathies, and to get a better understanding of host response and gut microbiota to different diets, in an intent to use the diet as a lever to re-equilibrate unbalanced functions, but also to determine or have a better understanding of individual variability of the response to diets. This in order, obviously, to increase diet effectiveness against chronic canine enteropathies. And our final goal will be to manage to reach a more personalized diet for semi-stray dogs or animal companions in general. So finally, I would like to thank you, all the members of the MIA team and all the collaborators that were together with us in these first studies. And thank you for your attention.